here. Okay. A second. Okay, now it's working. So let me start by introducing the group Blank. Professor Dr. Lars Blank is the head of the Institute of Applied Microbiology at the RWTH Aachen University in Germany. His research activities include biocatalysis, metabolic engineering, process intensification, and systems analysis. Recently, Jana Fees joined us as a new PhD student on Polyp research. A few words to my own person. I did a bachelor's in biology and a master's in microbiology at the University of Bonn in Germany. After that, I worked as an application specialist at Air Biopharm in Darmstadt, Germany. After that, I joined the group Blank to do my PhD, which I finished in February of this year. And now I'm working as a postdoc at group Blank. I am funded by the BMBF and the DBU. Many people contributed to our Polyp research activities, which I'd like to mention here. These are our project partners, namely Budenheim, with Dr. Rainer Schnee, Dr. Daniel Buchholz, Dr. Sven Bach, Dr. André Lemke. Then the group of Professor Dr. Ulrich Schwaneberg, together with Dr. Anna Joel Ruff and Kevin Herrmann, also David Schönauer and Kerstin Kardinier. Many thanks also to Professor Dr. Stephanie Smith, Professor Dr. James Morrissey from the University of Michigan, and to Dr. Sabine Wilbold from the Research Center in Jülich. Those people contributed to our publications on Polyp research. Also thanks to Jürgen Lamm and Eva Vetter from Algrano, Dr. Bernd Weinreich and Bajo Bajovic from Leiber, and Dr. Michael Quanz and Dr. Eric Pollmann from the Research Institute for Baker's Yeast. Let's start with a short introduction about polyphosphate. We have elemental phosphorus, then there is orthophosphate or short phosphate, and then there is inorganic polyphosphate, which is the polymer of phosphate. We differentiate three different molecular structures. First of all, the linear form, the cyclic form, and the branched form. The branched form is not stable in aqueous solution. The branching points spontaneously hydrolyze upon contact with water. The cyclic and the linear form are both stable in water, but industrially relevant is only the linear polyp. And also, I believe that most microorganisms and living beings produce solely the linear form of polyphosphate. So it is found in most living beings. The chain lengths can range from 2 to over 1,000 p subunits in living beings. The chemical synthesis allows only chain lengths from 2 to 40 on large scale. And the chain lengths, except for polyp 2 and polyp 3, is usually polydispersed, which you can see on this nice polyacrylamide gel electrophoresis picture, which I will discuss later in this talk. The alkaline metal polyphosphates, such as sodium and potassium, are water-soluble, whereas the Earth's alkaline metal polyphosphates, such as magnesium and calcium, are water-insoluble. So what do we do at Group Blank in terms of Polyp research? These are basically two topics. First of all, we develop polyphosphate analytics, and then we develop biotechnological Polyp production with Saccharomyces cerevisiae from unused phosphate waste streams. So let's start with the polyphosphate analytics. There are many parameters of interest in polyphosphate analytics. It usually starts with polyphosphate as an intracellular product. In this case, this is supposed to be a Baker's yeast cell. One parameter is immediately of interest, that is the cellular localization. For many other parameters, you need to extract the polyphosphate from the yeast cell. And then you can measure the length distribution, the cation composition, the total polyp concentration, the average polyp chain length, and the polyp we wrote a mini review called Methods for the Analysis of Polyphosphate in the Life Sciences. This mini review deals with analytical methods in polyp research. And what we did here can be seen in the key table from this publication. We listed all the relevant analytical methods in polyp research. And then we looked at which parameter these methods can measure. So the 6 parameters I talked about on the last slide are listed here. And then we looked at which are covered and if they are measured quantitatively or qualitatively. After that, we categorized those methods even further by a few criteria. First of all, if a prior polyp extraction is required, the polyp specificity of the method, the covered polyp chain length range, and the required polyp purity. And what you can do here is you either look at what you want to measure, for example, total polyp, and then you can go down and look at which methods are capable of doing so, or you look into your laboratory and see what kind of tools or machines you have, and then you can go to the right and then can check what you can actually measure with it. How do we do the polyphosphate analytics in our lab? We're starting again with polyphosphate as an intracellular product in Saccharomyces cerevisiae. First, we do an analytical polyphosphate extraction to get the polyphosphate out of the cell. And then we quantify the total polyp concentration and measure the average polyp chain length. And I'm going to talk about all those 3 methods and the colorimetric phosphate detection reagent on the following slides. The goal of an analytical polyp extraction is to get out all the polyp from Saccharomyces cerevisiae in a gentle way. On purpose, we omitted polyp silica purification and polyp precipitation with ethanol and barium. We did that because those purification steps neither work at low polyphosphate concentration, nor do they work quantitatively. What we actually did is we compared the most promising literature protocols, and here you can see the results. I'm not sure if this window is blocking the view. I'll pull it down. Okay, so here you can see the results. On the x-axis, there are 6 extraction protocols from literature, and on the y-axis, there is the polyp content in the Saccharomyces cerevisiae cell dry weight in %. So we produced a batch of polyp-rich S. cerevisiae, extracted it with 6 different protocols, and then measured the total polyp concentration. As you can see, depending on which protocol you use, you get quite different results. Okay, next, we optimized the best literature protocol, which was obviously the one on the right. This is from Brewer et al. 2016. And what we did, to summarize, we omitted the ethanol precipitation step, we omitted the chloroform washing step and the nuclease treatment, and then we did some step-by-step process optimization, and overall we were able to improve the extraction efficiency from here, the original protocol, to the optimized protocol by 40%. And we also simplified the protocol, which now requires only 1 tube and takes 30 minutes. The protocol is straightforward. You take 25 milligrams of cell wet weight, you add the buffer, phenol, and then you incubate for 10 minutes at 45 degrees. You add your chloroform and the phase lock gel, and then you centrifuge. Here's a picture of what you get. In red is the water phase, then there's the phase lock gel in between and the phenol chloroform at the bottom. The phase lock gel tremendously helps to take out all the water at the top. And this is actually just regular vacuum grease, which everyone has in its laboratory. It's super cheap and it works great. The question here is whether this extraction protocol is applicable to other species because we validated it only for Saccharomyces cerevisiae. Okay, now let's talk about the colorimetric phosphate detection. This is required for the enzymatic total poly-P assay I'm going to talk about on the next slide. So the principle is always the same. You take a sample containing phosphate, you add 1 to 2 reagents, a color develops, whereas the color intensity is proportional to the phosphate concentration in the sample, and then you read the absorbance. Many literature protocols use the malachite green assay. It has a very high sensitivity, defined here as the slope of the calibration curve. The problem here is that the blank signal takes at least 20 minutes for stabilization, which you can see in this graphic. On the x-axis, you have the time from 0 to 30 minutes, and on the y-axis, the absorbance. And this is just water plus the 2 reagents required for the malachite green assay. And it takes 20 minutes for the signal, so this is just a blank, to stabilize. Also, this assay requires 2 reagents. We went on to look at the molybdate antimony assay. It has a lower sensitivity, so only 7 times 10 to the power of -3 absorbance units per micromolar phosphate in comparison to 33. But the color develops super quickly. So it takes shorter than 2 minutes at room temperature for full color development, which you can see here. On the x-axis, the time from 0 to 10 minutes. On the y-axis, again, the absorbance. And we tested the blank and 3 different phosphate concentrations. And it took me about a minute to transfer the microtiter plate into the plate reader. And up to this point, the color was already fully developed and stable for more than 10 minutes afterwards. It requires a low acidity in comparison to the malachite green assay of only 150 millimolar sulfuric acid. And just to give you a reference, poly-P2 hydrolyzes at 1.3 micromolar per minute in these reaction conditions. What I failed to mention in the beginning is that also the malachite green assay happens under very acidic conditions. And the issue here is if you have low pH, poly-P spontaneously hydrolyzes, and this leads to a high blank reading in the total poly-P assay. Therefore, if you incubate for 20 or 30 minutes your phosphate assay, you artificially raise your blank measurement, and by that you artificially lower your actually reading in the sample, because you of course have to subtract the blank reading. So it is desirable to have a very quickly developing phosphate detection reagent. Okay, it's only 1 reagent, so it's more convenient in the laboratory. The linear range spans from 5 to 200 micromolar phosphate. Here you can see a calibration curve on the x-axis from 0 to 200, the phosphate concentration in micromolar, and on the y-axis the absorbance. And as you can see, there's quite a nice fit. Okay, now let's talk about the determination of the total polyphosphate concentration. We're starting with a transparent 96 well plate containing 100 microliters of sample. The sample contains poly-P. We are adding 2 enzymes, which are the PPX enzyme and IPP, which are the exopolyphosphatase and the inorganic pyrophosphatase from Baker's yeast. These are contained in 50 microliters of enzyme reaction buffer. This is just a schematic drawing, which will be animated in a second. Okay, but first, let's talk about the first reaction. PPX cleaves off all phosphate residues and leaves per poly-P chain 1 poly-P2. The poly-P2 is then further hydrolyzed into orthophosphate by IPP. And I hope this animation works, so you can see it. So PPX is attacking the poly-P chain, but it will leave 1 poly-P2, which is then hydrolyzed by IPP1P. Okay, and next you add 50 microliters of the phosphate detection reagent. I've talked about that on the previous slide. And now the whole thing turns blue and you can read the absorbance. In the laboratory, this looks something like that. So you add it, and then it quickly turns blue. Okay, now let's talk about the average poly-P chain length assay. So this is a bit more complex. So I'm going to give you a quick definition and example first. If you were talking to a chemist, he would call this parameter the number average degree of polymerization. In our laboratory, we just say it's the average poly-P chain length, and we define it by taking the total poly-P concentration and dividing it by the poly-P chain concentration. To give you an example, these are 3 poly-P chains. So the poly-P chain concentration is 3, and the total poly-P concentration amounts to 17. So you take 17 divided by 3, and then you get an average poly-P chain length of 5.6 P subunits. And how do you actually measure that in the laboratory? To measure the average poly-P chain length, you have to quantify the total poly-P concentration. This is what we talked about 2 slides before. And you have to measure the chain concentration. Okay, this is done in a black 96 well plate. You add 100 microliters of poly-P sample, and then you add 50 microliters of an enzyme substrate cocktail. It contains PPX, ATP sulfurylase, hexokinase, and glucose 6-phosphate dehydrogenase, and all those 3 substrates listed here. The first reaction is the same as on the left side. So per poly-P chain, PPX leaves 1 poly-P2. This is critical for this whole analytical procedure. The poly-P2 reacts then to ATP, catalyzed by ATP sulfurylase. ATP reacts with glucose, forming glucose 6-phosphate, catalyzed by hexokinase, and the glucose 6-phosphate is then reacted to NADPH by glucose 6-phosphate dehydrogenase. So what you have to remember here is that per chain poly-P, you get 1 NADPH in the end. And this you can quantify by reading the fluorescence. Of course, we validated the performance of this enzyme assay for the quantification of the average poly-P chain length by comparing it to 31 phosphorus nuclear magnetic resonance. We took a few poly-P chain length standards ranging from 2 to 270 P subunits. And then we quantified the average poly-P chain length by 31 P NMR on the x-axis and by our enzyme assay on the y-axis. And as you can see, both methods agree well. And what one also should mention is that the covered range is quite wide. So from 2 to 270, that's quite the range. Unfortunately, we did not have any longer poly-P, but I think it is possible to quantify even longer polyphosphates. To summarize the performance of the total poly-P assay and the average poly-P chain length assay, you can measure the reproducibility of the assay by taking one sample and measuring it repeatedly. And then you can calculate the relative standard deviation, which is quite low. It's 1.5% for the total poly-P assay and 2% for the average poly-P chain length assay. There is no interference from sodium chloride, sodium sulfate, ADP, DNA, and RNA. Especially these nucleic acids are important because they are oftentimes co-purified with the poly-P. There's no interference up to 4 grams of nucleic acid per gram of poly-P. If you have more, you have to degrade it prior to analysis. Both methods are capable of high throughput analysis, and you can measure down to micromolar poly-P concentrations. The required standards are pretty straightforward. For the total poly-P assay, you need a potassium phosphate standard. And the chain length assay consists out of the total poly-P determination, which is the same, and out of the chain concentration assay, which requires poly-P2 standards. But you do not need any chain length standards because the chain length is determined absolutely. The analytical tests are low cost and they require minimal equipment. For example, a plate reader and a multi-channel pipette would be nice. One disadvantage is that 2 of the required enzymes are commercially not available, so they have to be expressed in E. coli in the laboratory. Okay, let's summarize the analytics research at Group Blank. We developed a 1 reagent, 2-minute phosphate detection reagent, and an analytical poly P extraction for S. cerevisia. And then we developed 2 enzymatic assays, an in vitro total poly P assay and an in vitro average poly P chain links assay. So let's talk now about the biotechnological poly P production. So first, let's answer the question, what can you actually do with polyphosphate? The applications of polyphosphate are directly linked to the physicochemical properties of polyphosphate, which are listed here. For example, polyphosphate contains phosphorus, it's a mild acid, it's a soluble cation exchanger, and those properties, some of them are constant with the chain links, and some of them are more pronounced with short chain, and some are more pronounced with long chain. The applications, there are many of them. Our lab focuses on the production of food-grade polyphosphates. The application is seen, for example, in meat products. For example, in doner kebab, you can imagine on the outside it's quite hot, and then the inside it's frozen, and somewhere in between it has a nice 20 to 30 degrees, which is optimal for microbial growth. And here you need the bacteriostatic properties of poly P to conserve the doner kebab. Or in the production of soft cheese, you need polyphosphate as a soluble cation exchanger to complex the calcium from the milk. Now let's talk about classical polyphosphate synthesis. So the chemical route, it all starts with phosphate rock. This is our fossil phosphate reserve. This can be found as hydroxyapatite or fluoroapatite. And then there are basically 2 processes to purify the phosphate into phosphoric acid from the phosphate rock. The most dominant mining process is called the wet chemical process, where you add sulfuric acid to the phosphate rock and get a side product, which is called gypsum. This is calcium sulfate, and you get crude phosphoric acid, which has to be extensively purified to get food-grade phosphoric acid. And the other process is called the furnace process, where you mix phosphate rock with coal and silica, and then you heat the whole mixture. And what happens here is that the phosphate is reduced to elemental phosphorus and then distilled. So you end up with elemental phosphorus, also known as white phosphorus, P4, which is burned in an oxygen atmosphere. So you get phosphorus pentoxide. Of course, if you add excess of water to phosphorus pentoxide, you also get phosphoric acid. And now if you want to produce polyphosphate, there are 2 syntheses. The first one is called the condensation synthesis. So from here on below, I try to be stoichiometrically correct. And this is just an example to produce pyrophosphate. If you want to produce longer chain poly P, this will look differently. So you're starting with phosphoric acid. You add sodium hydroxide. This sets the counter ion of the poly P and the pH. You get as an intermediate disodium hydrogen phosphate and some water. And this mixture is then heated at 200 to 600 degrees for several hours. You want to get rid of those 4 water molecules and 1 more to join the 2 phosphate molecules, forming sodium pyrophosphate. The other synthesis is called the hydration synthesis, where you take phosphorus pentoxide, you add ammonia and water. This is a limited amount of water. If you add an excess of water, you get phosphoric acid. The ammonia sets the counter ion and the pH, and this initiates the hydration reaction, and you end up with ammonium pyrophosphate. This synthesis route is suitable for counter ions which are volatile. To summarize, we're starting with fossil phosphate rock, which is mined into phosphoric acid, which is mostly used as fertilizer. This ends up in the soil, is washed out into rivers, and then washed out into the ocean. the natural reformation of fossil phosphate rock takes 10 to 100 million years. And of course, this natural reformation rate is way slower than what we currently mine. The phosphoric acid used in food products ends up in sewage sludge. Currently in Germany, this is still applied to farmland, but starting in 2029, it will be mandatory to directly recycle the phosphate either from the sewage sludge or from the sewage sludge ash. There are a few issues associated with the phosphate rock mining. First of all, that's the uneven global distribution. If you look at this graph, you can see that only a few countries worldwide even have phosphate rock. And most of the phosphate rock, 73%, can be found in Morocco and the Western Sahara. Then there's the limited nature of phosphate rock. We have about 70,000 megatons of phosphate rock on our world. And we are currently mining 270 megatons per year. If you now look country by country, those are the 23 countries worldwide which have their own phosphate rock reserves. And if you now calculate the number of years for which the national phosphate rock reserves will last, so if you just extrapolate the current mining rate of each country, you can see when the phosphate rock reserves for this country will be gone. Let's look at China, India, and the United States. Together, they inhabit 40% of the global population, and their phosphate rock reserves will be gone in under 40 years. If you look at the global average in red here, you can see that our global phosphate rock reserves will be gone in under 300 years. Then there's the transportation issue because most is found in Morocco. Then the environmental destruction and pollution during phosphate rock mining, and at last the contamination of phosphate rock with toxic and radioactive elements. To give you an idea, for the use in fertilizer, it is economically not feasible to remove those toxic and radioactive elements. And what you end up with is about 100 milligrams of uranium per kilogram fertilizer. And this leads to an input of about 10 grams of uranium per hectare farmland per year in Germany. Our strategy therefore must be to reduce, reuse, and recycle phosphate. And here biotechnological poly-P production comes into play. We're starting with unused phosphate side or waste streams. For example, this is canola press cake. So if you take the canola seed, press out the oil, the canola seed press cake is what remains. And this we did together with project partners, but this is just one example for an unused phosphate waste streams. We are looking to use even more. Here you can see the poly-P metabolism in Saccharomyces cerevisiae. I'm not going too much into detail here, but what you should note is that low phosphate concentrations in the cell trigger the Faux pathway, which can be seen here. And the Faux pathway triggers the expression of, for example, high affinity phosphate importers, and also the vacuolar transporter chaperone, which actually produces a polyphosphate. There are several advantages from biotechnological polyphosphate production. First of all, you're independent from phosphate rock imports, and the phosphate source itself is cheap and accessible. From an environmental standpoint, there is no more polluting phosphate rock mining. And looking at the product quality, you're able to produce chain lengths above 40, which is not possible on large-scale chemical synthesis. You have this organic label. In Germany, this is called bio, and the downstream process is tailor-made. So you can easily switch the counter-ion, chain length, and purity, which is not easily done in chemical synthesis. How do we do this? The biotechnological poly-P production. We are starting with wild-type S. cerevisiae. We first put this S. cerevisiae in a medium which has no phosphate, but all other components required for growth. This is called phosphate starvation. And then we transfer the starved cells into a medium containing phosphate. The cells then take up more phosphate than they would usually hold. This fat S. cerevisiae can be used to either produce a poly-P-rich yeast extract or a pure biopolyphosphate. The starvation and feeding step together are called polyphosphate hyperaccumulation which I'm going to talk about on this slide. Just a few key features. Our production host is a wild-type S. cerevisiae. This is generally regarded as safe, and it's a non-GMO. We can achieve high titers of 28% poly-P, SKPU3, and cell dry weight. The kinetics are shown here. On the x-axis is the time from 0 to 6 hours, that is the starvation step, and from 0 to 3 hours, that is the feeding step. And on the y-axis is the polyphosphate, SKPU3, and Saccharomyces cerevisiae cell dry weight in percent. Normal cells contain about 2% polyphosphate, which goes down to 0.1% polyphosphate in cell dry weight. And during the feeding, within 2.5 hours, the polyphosphate content goes up to 28% in cell dry weight, which is remarkable. Here's some more performance parameters from the fermentation. The titer can be expressed as well in 2.7 grams poly-P per liter, so a different unit. The rate is about 1 gram poly-P per liter per hour, and the yield defined as mole poly-P recovered divided by mole phosphate in the feeding medium amounts to 0.39, and this can go up to 0.75 if you reduce the phosphate concentration in the feeding medium, which decreases only slightly the titer. No aeration is required, and the phosphate source is variable. Of course, there's potential for optimization. We want to increase the cell density by switching from a batch to fed batch fermentation. And we want to switch the 3-pot fermentation, which includes the cell mass production, starvation, and feeding, to a 1-pot fermentation. And we want to switch the carbon source from glucose to molasses. Now let's talk about the preparation of pure biopolyphosphate. So from the fat cells to the pure biopoly-P. How do we do that? This is supposed to be a cell envelope of Baker's yeast. We have the nucleus with DNA, the endoplasmatic reticulum with some RNA, and the vacuole with short chain and long chain polyphosphate. The frozen cells are suspended in water. This is one freeze-thaw cycle, and then the cells are heat treated for 10 minutes at 70 degrees. This opens up the cells, and then the water-insoluble debris can be removed by centrifugation. Next, we add HCl to lower the pH, and what happens here is that the nucleic acid and the proteins get protonated and thus water insoluble and can be removed by centrifugation. Okay, next we set the pH. Interestingly, the pH we are setting at this step is kept by the polyphosphate throughout all the remaining process steps. This means we want a pH neutral final product, therefore we set the pH here at a neutral value. Next we add some sodium chloride and a little bit of ethanol. So what is happening here is the sodium is neutralizing the charge of the polyphosphate, and by just adding a little bit of ethanol we are capable of precipitating only the long chain poly-P, which then can be recovered by centrifugation. After that we add some more ethanol to also recover the short chain polyphosphate. Next, we want to wash our polyphosphate pellets with 50% ethanol to get rid of salt residues. And afterwards, we dry the polyphosphate pellets for 7 days in a desiccator. Of course, on an industrial scale, this would be done with a different drying method. The last step is the milling. This is currently done with a ball beater. This is the starting material, very coarse, and this is a fine-grained final biopolyphosphate. So this is how we produce biopoly-P. We are able to analyze the product properties in detail, and I want to show you our findings here. We are comparing a chemically produced sodium polyphosphate called Wooded-4 with a sodium long chain and a sodium short chain biopolyphosphate. All three appear as a fine-grained white powder. The molecular formula can be seen here. The chemically produced poly-P has only sodium as a counterion, And the biopolyps have an equal molar ratio of sodium, potassium, and magnesium as counterions. So in a way, the name sodium biopolyp is a bit misleading. But of course, in the end, we want to get rid of the potassium and the magnesium. The Butyl-4 dissolves quickly within 5 minutes, and the biopolyp takes its time, about 30 minutes to dissolve, but then fully dissolves. This is due to the magnesium, which slows down the dissolution. The molecular weight is derived from the molecular formula, and the distribution of the chain links can be seen on this polyacrylamide gel electrophoresis picture, which we already saw in the beginning of the presentation. On lane 1, you have the chain links ladder. Lanes 2 and 3 show independent batches of long chain sodium biopolyp. Lanes 4 and 5 show short chain sodium biopolyp, and the lane 10 shows the Butyl-4. Unfortunately, this method is not able to measure polyp which is shorter than 15 p subunits. The chain links distribution of the long chain sodium biopolyp ranges from below 15 to about 300 p subunits. The short chain variant ranges from below 15 to 130 p subunits, and the Butyl-4 ranges from below 15 p subunits to about 200 p subunits. The pH, bit typical, is very low for the chem polyp, and the pH of the biopolyps is neutral, which is desirable and which we can set during the downstream processing. The crystal water content is very low in the chem polyp, which would be expected because the production involves heating at several hundred degrees. And in the biopolyp production, water is removed by drying in a desiccator, and apparently this desiccator is not capable of pulling more water. So we have about 6% of crystal water. The average polyp chain links amounts to 19 in the chem polyp, 11 and 42 in the short and long chain biopolyp respectively. The purity of the chem polyp amounts to 89%. We also quantified 2 other chemically produced polyphosphates, and there the purity was as low as 85%, and the purity of the biopolyps was between 85 and 91%. So the purity of the bio and the chem polyp was on the same level. All 3 are fully water soluble, so no insolubles detected. There was no chloride detected. This is important because sodium chloride is used as the precipitation agent, and itself did not precipitate. Interesting difference between the chem and biopolyp is the cyclic polyp content. The chem polyp contains around 5 to 10% cyclic polyp, whereas the biopolyp contains 2% or less cyclic polyp. So, and this is another argument for the fact that Cerevisia is producing only linear polyp. Phosphate was only detected in the short chain variant, and nucleic acid were detected at below 2%. This is important because nucleic acids behave somewhat similar to polyphosphate in the downstream processing and have to be removed by the HCl precipitation step. All these elements were not detected, which is beneficial because we are seeing the application in food products. To summarize, we developed a fermentation protocol to produce Cerevisia with a very high titer of 28% polyp. From this polyp-rich yeast mass, we can produce food-grade polyp-rich yeast extract, or we can produce food-grade pure biopolyp. We are always looking for collaborations. For example, we can do polyphosphate analytics for other groups. We are interested in phosphate recycling and the application of our biopolyp, but we are also always open for suggestions. If you're interested, please contact Lars or myself. Thank you all for your attention. Again, special thanks to Patrick for making this webinar possible. And if you have questions, feel free to ask.
Okay, so we'll go ahead and open things up for questions. Are you able to see them, Jonas?
Yes. Okay, so the first question is from Mike Downey. Very cool stuff. Is there evidence for poly-P chains attached to or kept by other small molecules in vivo? Yeah, the thing is that we wouldn't even see that with our analytics because the total poly-P assay involves the action of the PPX enzyme, which degrades the poly-P down. And if there would be an end cap, the PPX wouldn't be able to hydrolyze the poly-P chain. So we are not able to see this. This would only be possible, I guess, with nuclear magnetic resonance measurements. Okay, another question. Is there evidence for cyclic poly-P in vivo? Yeah, good question. So we haven't found cyclic poly-P, and we are actually pretty happy about that because industrially relevant is mostly only the linear form. And from literature, I know that linear poly-P is like most dominant in living organisms, but I think there are a few publications out there where cyclic poly-P has a function in vivo. So I'm going to be a bit precautious on this topic. Okay, Michael Gray asked, have you tried your analytical poly-P extraction and chain length assay on any bacterial cultures? No, we have not. So we're working solely with Baker's yeast, but we are always happy to do some collaborations where you can send us samples and we do the extraction and the analysis. And it would be very interesting to see how results differ to other extraction protocols. Okay, Robert De Campo asks, very nice talk. Do you consider short chain poly-P up to 130 mer? Yeah, okay, that's question of definition. So we are mostly looking at the average poly-P chain length, and in our, let's call it short chain variant, the average chain length is 11, if I remember correctly. And from our standpoint, this is considered short. Keep in mind that the chemical synthesis is able to produce something from 2 to 40 p subunits. 2 to 40 p subunits, so 40 would be considered long and 2 would be considered short. So 11 would be either intermediate or short chain, but this all depends on the definition. And you're right, on the polyacrylamide gel electrophoresis, you can see that there's quite the wide mixture of polyphosphate chains. Okay, Dieter Jendrusek asks, why is cyclic poly-P stable in water, but branch poly-P is not? The thing here is that it's thermodynamics or something. So if on a poly-P chain, there's like another attachment, this is super unstable and just hydrolyzes. And this is shown in literature that it's just not stable in aqueous solution. If you're looking at solid poly-P, this is stable. But if you're talking about microorganisms where the poly-P is considered to be in solution, we do not consider branch poly-P to be in the Baker's yeast. Okay, how much poly-P do you lose in the bio poly-P preparation from the acid step? Asks Richard Gomer. Good question. So what you're referring to is the extraction efficiency. And if you're comparing the analytical extraction to our preparative extraction, then this is 80%. So we get 80% of what we would get with the analytical extraction. So you lose about 20%. The thing is that you have to always be, you always have to look at the economics of the process, because in the end, this has to be an industrial process, which makes at least a little bit of money. So we cannot do as many steps or as many expensive steps as in the analytical extraction. Andreas Meyer asks, very nice, how does the cost of biopoly-P in yeast or after purification compare to that of chemically prepared poly-P? Any idea? Yeah. So if you're looking at food grade polyphosphate, if I remember, the price is somewhat between 1 and 5 euros per kilogram. And we are at least a factor of 10, rather a factor of 100 away from that. So there's still some optimization required to make this process economically feasible. Ramesh Rijal asks, how hard would it be to extract poly-P from cellular organelles? I don't know. So we're biotechnologists. So we are concerned with biotechnology. We just look at all the poly P we can get with a certain method, and we do not look at where it comes from. So whether it's in the vacuole or in the cytosol or attached to the cell envelope. In the end, we just want our product with good product quality in the end. So you would have to test that on your own. Okay, Samantha Ganga Persat asks, why are you changing the carbon source from glucose to molasses? Yeah, this is normal to do that in yeast industry. For example, if you're going to the supermarket to prepare some pizza and you buy your little cube of baker's yeast or even the dried baker's yeast, this is all produced with molasses because molasses is very cheap. It's a side product from the sugar industry. Rashna Bandari asks, would your assay measure poly-P covalently attached to proteins? So you have to specify which assay we are talking about. So it's the total poly-P or the average chain length. The total poly-P assay would work as long as one end of the poly-P is free for the PPX to attack. So if one end is attached to the protein, the other end can be attacked by the PPX, can be hydrolyzed, and the released phosphate can then be quantified. The average poly-P chain length assay would not work because there we need the poly-P2, which is left over from the whole poly-P chain after PPX did its work. Felix Ruiz asks, nice talk. Any idea of how we could improve the sensitivity of the colorimetric poly-P measurements? Okay, so if you say sensitivity, you mean the slope. So because the limit of detection is not a problem because we have to dilute our samples anyway. But actually I have no idea how to improve the sensitivity. This is just, I guess you cannot change that. That's quite common for these molybdate phosphate assays. Rodrigo asks, do you have an idea of how long is this biopoly-P stable in water after solubilization? This depends. So polyphosphate spontaneously hydrolyzes upon contact with acid, alkali, magnesium, enzymes, high ionic strengths, and temperature. So if you're being gentle to your poly-P, so think demineralized water, neutral pH, no magnesium, no enzymes, no acid, no alkali, no salts, then this is super stable. But if you're adding any of those components, then the poly-P will be degraded. Also, this magnesium content seems to be interesting to make solid gel materials for bone regeneration, for example, right? Yeah, good point. We have not yet tested any applications of our biopoly-P. So if you are doing that in your laboratory and need maybe only a few grams because it's hard to produce much more in our laboratory, just contact us. We will send you some samples and then you can test it. Okay, I guess these were all questions.
All right, so that was a fantastic talk. Thanks so much, Jonas, for joining us. And then just a reminder to everybody that we'll be having the social hour in about 10 minutes from now. So I encourage anybody to check that out if you're interested. And then next week, we'll be having Werner Mueller and Zhao Hong give a talk. So I recommend checking that one out as well. It should be fantastic. So thanks everyone for being here. And thank you, Jonas, as well. And I'll see all of you next week. Thanks.
Bye.